Good afternoon, students. Today I've decided to take a little bit of time to go over some strategies for quiz and test taken in Biology 10. However, I want you to know that these strategies can be applied to any class that you take. Specifically, we're talking about how to take multiple choice quizzes and tests more successfully. Because I've noticed overall the average on the last quiz was not bad. But I noticed that some students appear to be struggling, and some of their answer choices will lead me to believe that some folks really don't have a clue when it comes to taking multiple choice quizzes and tests. So the purpose of this video is to review some of the more successful strategies to use that can help you even if you're not even sure what the answer to a question is, but using the process of elimination you'll more than likely guess the question correctly. Alright, you recognize this. This is the last quiz we took. What I'm going to do is take the quiz right now, and I'm going to go over with you the technique that I use for selecting the correct questions, even if I wasn't the biology instructor. Alright. Okay, you recognize this window here. Alright. Let's take a look at this question here. How are these two amino acids attached together? Alright, we have an amino acid here on the left, an amino acid here on the right. And that's the equation. The reactants here are two amino acids. The reaction is a dehydration reaction, which yields water and a dipeptide. Well, take a look at here. They've highlighted the hydroxyl group on this amino acid and this hydrogen on this amino acid. Notice that this OH is attached to a carboxyl group on one end, whereas this hydrogen is going to come off this amino group on the other end. Let's read through the choices and let's see which choice is the most correct. Now the first one is amino group to amino group. Well, no. That's not the answer. The second choice is amino group to carboxylic acid group. That's the correct choice. But let's go through all the groups to see if there's one choice more correct than that one. The third choice is through a hydrolysis reaction. False. It's a dehydration reaction. Question gave you that. See, water is yielding. And the last choice is carboxylic acid group to carboxylic acid group. Well, you can see right here that's incorrect. So the answer is number B, or number 2, amino group to carboxylic acid group. Alright, let's scroll down here. This next question is asking you about peptide bonds. What do peptide bonds do? Well, they join amino acids together. Well, let's go through the choices here. First one is link amino acids. That seems to be the correct choice. Are used to form amino acids? No. Amino acids are already formed before they're joined by peptide bonds. Formed between fatty acids? No, that's very false. Are formed by a hydrolysis reaction? False. It's a dehydration reaction. We saw that in the first question. So the first correct answer is the first one. Linked amino acids. Alright. Now, these two molecules are structural isomers. What is the difference between them? Well, do you know what a structural isomer is? You have to know that first. And simply put, it's two different molecules that have the same molecular formula. Glucose has the molecular formula of C6H12O6. Well, fructose does also, C6H12O6, but the difference is the arrangement of the carbonyl group, the arrangement of the atoms. On the aldose, the carbonyl group is here, but in the ketose, it's in the middle. So what is the difference between them? the location of a double bonded oxygen atom? Yeah, that seems to be the correct choice. But I'm going to go through all the choices. The number of hydrogen atoms, that's false. They both have the same number of hydrogen atoms, so that's not the difference. Number of carbon atoms, false. They both have the same number of carbon atoms. And only one of them has a double bond between carbon atoms. All the bonds between the carbons are single. The only double bond is between the oxygen and the carbon. The carbonyl is here. So the location of the double bonded oxygen atom is the correct choice. So I marked that one. Question number 4 is, which of the following foods would be least likely to contribute to the development of coronary heart disease? One of the things I've seen is that people are not reading the questions correctly. They don't understand what the question's asking for. So, which of the following foods would be least likely? Well, margarine, bacon, fish oil, and butter. Well, butter is full of saturated fat. Saturated fats are correlated with coronary heart disease, along with bacon. Margarines contain trans fats, which are unsaturated, but they behave like saturated fats. Fish oil is made from omega-3 unsaturated fatty acids. Fish oil that's the correct choice. Alright, okay, next question is that lactose intolerance is the inability to what? It's an inability to digest lactose. People that are lactose intolerant cannot drink milk or consume dairy products that have milk in them because of the inability to digest the lactose. Well, let's look through the choices. They produce lactose? No. Digest cellulose? No. Produce milk proteins? No, none of those make sense. But digest lactose, that's the correct choice. And question number 6 is asking you about what is the storage form of carbohydrates in animals and in plants. Well, it's glycogen in animals and starch in plants. So glycogen, cellulose, no. Cellulose, glycogen, no. Starch, glycogen, that's the reverse of the correct choice. It's number 4, glycogen in animals, starch in plants. Which of the following statements regarding nucleotides is false? Alright, there's a fault. 3 of those statements are going to be true. 1 is false, select the false one. Nucleotides can be linked together to form nucleic acids. That's a true statement. Nucleotides contain lipids. False. Now, I'm not going to mark it yet though. I'm going to go through the other 2, even though I know that's the correct one. Nucleotides contain nitrogenous bases. That's a true statement. Nucleotides contain sugar molecules. Yes, they do. Pentose sugars. Ribose and RNA, deoxyribose and DNA. So, the false statement is number 2. Question 8. Cellulose differs from starch in that cellulose is highly branched, whereas starch is unbranched. Well, that's true. But is that the most correct choice? Starch is formed by plants, cellulose by animals. False. Cellulose is a plant product, not an animal product. Starch is made of glucose monomers. This is true, while cellulose is made of fructose monomers. False. Both are glucose monomers. And now here's the answer. Most animals cannot break down cellulose, whereas starch is easily digested. That is the most correct answer. So that's the answer I'm going with. That seems to me to be most correct in this choice. Alright. High fructose corn syrup is made from corn. Fructose is a monomer. It's a monosaccharide. The main carbohydrate in corn is a polysaccharide called. Well, there's only one polysaccharide found in corn kernels, and it's not cellulose. Now, some people were choosing fructose, but fructose is a monosaccharide. A hydrocarbon is not a carbohydrate. Starch is. That is the correct answer. Okay. Now, what is the complementary sequence to the DNA strand TCGATGG? You know, this is a case where it'd be helpful to have a sheet of paper, a blank sheet of paper, and you could, and using the base pairing rules, you could write that out. Then using DNA base pairing rules, write out the complementary sequence. Well, the DNA base pairing rules are adenine with thymine, so A, C with G, guanine, G with C, A with T, T with A, G with C, G with C. Now, which choice matches that? Well, since this is multiple choice, we know it's going to start, the C complementary sequence is going to start with an A. So we immediately eliminate that one and that one. Alright, AG. Well, AG. Okay. G. So it's going to be a C. Well, they both have a C. We're not there yet. AT. So AGCT. Ah! We can already know that's the correct one because there's no U in DNA, only in RNA. So you don't have to take it any further. But there's an A here, there's a T there, so that matches up. There's a C there, G there, C there, G there. So that's why that's the correct choice. But you knew this was correct as soon as you saw that U there, because U is not in DNA, just in RNA. Alright. Question 11. Microfilaments differ from microtubules in that microfilaments are thicker than microtubules. False. Microfilaments are the smallest in terms of the cytoskeletal elements. Alright. Help to anchor organelles, whereas microtubules' primary function to help cells change shape and move. Let's go through the other choices. Are mainly composed of actin, whereas microtubules are composed of tubulin. That's a true statement. Is that the most correct statement? Are found only in plants, whereas microtubules are found in both plant and animals. False. That's false because the three types of cytoskeletal elements are found in all eukaryotes. So, and actually it's intermediate filaments that help to anchor organelles. And about all microtubules do is move the chromosomes around during cell division. So, I'm going with the third answer here. Yep. Alright. Which of the following statements regarding the Golgi apparatus is false? Alright, again, three statements are true. One is false. You got to choose the false one. Alright, the Golgi apparatus decreases in size when a cell increases its protein production. Why would it do that? It would not decrease its size. That is just think common sense would tell you that's false. If it's going to increase its protein production, it's going to increase in size, because what does the Golgi apparatus do? It processes proteins for transport, for secretion outside of the cell, or for other destinations inside the cell. Well, I bet you that's the correct answer. But let me go through the other three just to be careful here. The Golgi apparatus serves as a molecular warehouse and finishing factory. That's true. The Golgi apparatus modifies chemicals received from the endoplasmic reticulum. That's its job. That's its definition right there. The Golgi apparatus works closely with the endoplasmic reticulum. Very true. So, obviously, the first choice is the correct answer. That is a false statement. Alright, question 13. The endosymbiosis hypothesis proposes that two cells merge into one cell, improving the enzyme function of the new cell. False. Two separate cells work cooperatively, and one benefited from the other. Let's look through the other two before I rule that one out. A large cell engulfed and digested a smaller cell, exposes its enzymes for use by the larger cell. That's a false statement. So already we've ruled out number 1 and number 3. Throw those away. So now we got to look at number 2 and number 4, see which one is most correct. Let's look at number 4, which says a small cell lived inside a larger cell to the benefit of both cells. Well, that's the answer. If you understand the endosymbiosis hypothesis, that is the correct choice. Alright, number 14, Tay-Sachs disease. This is due to the absence of an enzyme that digests polysaccharides? No, that's false. Prevents the breakdown of glycogen? False. Causes an accumulation of lipids in brain cells? True, because it's a neurological disorder. Involves damage to liver cells? False. Go with number 3. Alright, now another thing I want to tell you. What if you get a question you don't quite know the answer to? You should not leave it blank, because you have a 1 in 4 chance. If you guess it correctly using the process of elimination, you can probably guess correctly. And that's better odds, folks, in Las Vegas. Alright, let's look at this question here. Let's pretend I don't know the answer. It's going to be hard, but I'll try. Which of the following statements regarding cells is false? All cells have a cell wall? No, that's definitely false, because animal cells do not have a cell wall. And certain members of the protists don't have cell walls either. Alright, that's definitely a false statement. Do we just mark that and ignore the rest? Look at the other choices. All cells can interconvert forms of energy. That's what happens in cellular respiration when food energy is converted into ATP. All cells have DNA as their genetic material. True. All cells are enclosed in a membrane that maintains internal conditions different from the surroundings. That's the plasma membrane. All cells have a plasma membrane. Alright, so the first one is correct. But I always read through all the choices before I choose one. And you need to too. I expect that. Question number 16. Alright, we got a chart, a graph, a diagram. Alright. What's this diagram telling us? Well, the distribution of protein inside a cell at the various stages of interphase. Gap 1, S, and gap 2. Well, let's read the statement. Much of the intracellular structure of a eukaryotic cell is involved in protein synthesis, especially cells that are dividing. The figure shows the amounts of protein in cells at different parts of the cell cycle between two cell divisions. Gap 1 is the stage after the cell has divided, and G2 or gap 2 is the stage just before the cell divides again. S is the stage when the cell is synthesizing materials such as DNA, mitochondria, and other organelles. Alright, that's a mouthful. It's a lot to consider. What is the graph? Okay, what's the graph telling us? Alright, that's the question. protein level in cells remain constant throughout the cell cycle? No, that's false. You can see how it goes up and down, up and down. At any given time, there are more cells in G2 than G1. There's nothing in that graph that indicates anything like that, so that's false. Not all cells at any given stage have the same amount of protein. That's about the only thing I could deduce from that. But let's look at the last ones. Protein used in G1 is recycled to produce protein in S. Well, whether or not it is or not, it's not. That graph is not telling you that. The only thing you can deduce from that graph is number 3. Not all cells at any given stage have the same amount of protein. That's why you pick that one. Learn to read the graphs. Not that difficult. A graph is 1,000 words. Alright, question 17. Cyanide inhibits mitochondrial function as a result. The rate of photosynthesis increase. That's got no. Got nothing. That's a well, forgive my English, a bullshit answer. Get rid of that. ATP synthesis increases. No, if you inhibit the mitochondria, it's going to decrease, which is the third choice. Protein synthesis increases, got nothing to do with that. The correct answer is the third choice. Question 18, which of the following statements about electron microscopes is true? Alright, let's read through the choices. Remember, one statement's true, the rest are false. Scan and electron microscopes are used to study the details of internal cell structure. That's false. That's what transmission electron microscopes do. Specimens must be sectioned to be viewed under a scan electron microscope. That's false. Only with a TEM. Okay, electron microscopes focus electron beams to create a magnified image of an object. Very true. That's why we picked that one. But before we actually mark it as the true statement, let's look at the last one. Okay? Transmission electron microscopes are mainly used to study cell surfaces. False. That's what a scanning electron microscope do. So the third choice is true. It is true, and it's the correct answer here. Alright, oops, there's that same graph again. Alright, I'm going to read it again. Much of the intracellular structure of a eukaryotic cell is involved in protein synthesis. The figure shows the amounts of protein in cells at different parts of the cell cycle between two cell divisions. G1 is the stage just after the cell has divided, and G2 is the stage just before the cell divides again. That's all during interphase. And S is the stage when the cell is synthesizing material such as DNA, mitochondria, and other organelles. Okay, blah, blah, blah. You've heard me. Alright. Most of the protein measured in the cells was likely synthesized by ribosomes. Well, that's their job. SMOOV-ER? No. SMOOV-ER synthesizes lipids. So that's incorrect. Nucleolus synthesizes the components for ribosomes, but that's not the correct choice. Mitochondria are the powerhouses. They produce ATP. So, ribosomes is the only correct choice. Don't let that likely, it was the ribosome. Don't let that likely throw you. Don't let the wording confuse you. As long as you know the material, don't let the wording confuse you. Read carefully. Make sure you understand it. But don't doubt yourself. And another thing, when you're taking a multiple choice test, once you make a guess, stay with it. Do not change your answer. Because very often what happens when students change answers? They mark the correct one first and then they change it to a false. 9 times out of 10, that's what's going to happen. So don't change it. Unless you're absolutely very sure. But if you doubt yourself, then once you mark it, leave it. All right. Last question. Okay, according to this figure, which of the following are large enough to see in the light microscope? I'm going to make all right, I want to make it a little smaller. Okay. The unaided eye is here. Okay, the light microscope, basically you can see frog eggs. Here we have plant and animal cells. Here we have bacterial cells. And actually, yes, you can actually see a mitochondrion with the light microscope. With using the special stain, you won't see much. You really need an electron microscope to see any significant details, but you can actually see them with a light microscope. Alright, and then here, this is all electron microscope territory. Alright, now I'll blow it back up. So, which of the following are large enough to see in the light microscope? Let's look at the choices. Mitochondria? Yes. Atoms? Right here? No. Molecules? Right here? No. Proteins? No. So you pick mitochondria. Alright. Now we'll submit the quiz. Once you're sure, go back through. Don't change anything. But make sure you answered all the questions. Alright? Make sure you answered all the questions. And from this point on, if you leave any question unanswered, I'm going to go back in and knock off an extra 2 points. Just because I can. Just kidding. I won't do that to you. I can't do that to you. I won't do that to you. I should, but I won't. But remember, leave nothing blank, because you're only shooting yourself in the foot if you do that. Alright? Alright. Yeah, we answered them. Okay, I'm running out of time here. But it was time well spent. I hope this helps you. Alright, I'll submit my quiz. Alright. Okay, well, 20 out of 20? Well, I know I'm the instructor, but still. You can do that too. The thing is, it's just not me. You too can earn 20 out of 20, you know. And I've had students that already have done that. Alright. This concludes the presentation. Enjoy the rest of your Labor Day and keep in touch with me. Bye-bye.